Okay, we're closing in on our third and last keynote address, which is given by Dr. Stephen Grupp, and I'm very pleased to introduce him. Dr. Grupp is the chief of the Cellular Therapy and Transplant Section, director of the Susan and Stephen Kelly Center for Cancer Immunotherapy, and medical director of the Cell and Gene Therapy Lab at CHOP, as well as the Novotny Professor of Pediatrics at the UPenn Perelman School of Medicine. Dr. Grupp came to CHOP in 1996 after receiving his MD PhD in Cincinnati and training in Boston. His primary area of research is the use of CAR T and other engineered cell therapies in pediatric cancers and other life-threatening disorders such as sickle cell disease. He led all of the pediatric ALL trials of CTL-019, including Novartis International Registration Trial, leading to the first FDA approval of a CAR product such as Kymriah. I butchered that. As a result of this work, he presented the clinical perspective at the first FDA ODAC CAR meeting. Dr. Grupp was elected to the National Academy of Medicine in 2019. Welcome.
Thank you very much. Actually, one of the great things about the cell and gene field is that if you thought the generic names of drugs were unpronounceable 5 years ago, you ain't seen nothing yet. So Kymriah is the trade name, and Tisagen-Lecklucel is the commercial name. And then there are commercial names that are coming up that I literally can't pronounce without practice. So I'm going to spend a quick 15 minutes really jumping off on what Kevin Mahoney was talking about this morning in terms of walking on the shoulders of giants. And so this is a quick anecdote on 10 years ago, 11 years ago, how we got started on the cell therapy journey, the cell part of cell and gene, engineered cell therapy, and putting the cell in Silicon Valley. And hopefully you've heard that term. It was actually coined, I believe, by Bruce Levine at Penn, and it really reflects the fact that cell and gene is just going nuts, and I'm already hearing that in the part of the meeting that I was privileged to hear, and apologies for arriving when I did, but I'm on call. And that little bag that you see at the bottom of the slide is Kymriah, and hopefully there's some discussion about cost issues. When that was FDA approved, it was FDA approved at $475,000, and that was kind of shocking, and I was directly involved with the development of what at that time was one of the most expensive single cost agents in medical history. But now, with what we see with Zolgensma at $2.1 million and Zynteglo at $2.8, that seems somewhat quaint. And hopefully we're not headed into an infinite cost spiral, because I'm not sure, to be honest with you, that the market is going to tolerate that. So I want to start, just for those of you who are interested in the notion of CAR T, chimeric antigen receptor T cells. This is the big part of cell therapy these days. Just wanted to show you what this looks like. And so what you have is two green cells. Those are ALL cells from a patient who died of ALL, completely drug resistant. And this is the experience that we have clinically. And then that gray cell down at the bottom is a T cell that you make from each individual patient, genetically engineer them. I won't go into the process, create these CAR T cells. And the CAR T cells come in, grab onto that ALL cell, and then that bubbling that you see is the destruction of the ALL cell. And the thing that makes this work is it's different from everything else. It ain't chemo, it ain't radiation, it ain't surgery, it's totally different. And therefore we can take cells that are completely resistant to all of the therapies and put these patients into remission and keep them there for years. So want to quickly go through this. So at University of Pennsylvania, Carl June, David Porter, Bruce Levine, the first patient with adult leukemia, CLL, was treated in 2010. And then we moved on to childhood ALL, treating our first patient in 2012. I'll tell you that story very briefly. It's not a, I heard the word slow about life science. I mean, it's not bad. Treated our first kid in 2012. Registration trial opens in 2015. This is now Novartis is in charge of this. Goes to the FDA AdCom in July 2017. All right, we're 5 years into the mission here. Not terrible. And then FDA approval happens a little bit later. And this was the first CAR T in kids. That's weird, just by accident mostly, because we made it work. First CAR T, first gene therapy in the United States. And now we got 6 CAR T products and 2 injected gene therapies, Zolgensma and Luxturna, which Luxturna, of course, is very familiar to a number of people in the audience. And this is the 10-year text from the Emily Whitehead Foundation, this young lady who we treated 10 years ago. And let me tell you briefly about her story. And I'm seeing from the slides that other folks are presenting that I need to clitzify my slide presentations. But 7-year-old girl, completely refractory ALL, was basically told to go home and get hospice. Our first patient came in with a ton of leukemia, which now we're learning that if you have a lot of leukemia, you have a lot of CAR T, and if you have a lot of CAR T, you get sick. But we learned how to fix it. And we learned how to fix it from her. She developed this life-threatening toxicity, which we now know a ton about, which is called cytokine release syndrome, or CRS, but we didn't know anything about back in the day. On the fly, in the moment, with the help of the ICU, with the help of the folks at Penn, developed a treatment strategy by repurposing a rheumatoid arthritis drug, tocilizumab, Actemra, that we call TOSI, for this purpose. Was not on the protocol, was not something that we imagined would actually happen, but we did it. And it really turned her case around from being critically ill in the ICU to getting better overnight, within hours. And so that is really the basis for 15,000 people getting CAR T across the world. This toxicity management process, these first patients at Penn, getting the whole ball rolling into this area of CAR T that we've heard so much about. Incidentally, I'm a transplant guy. That's what I've done my whole career. I got dragged into cell therapy sort of by accident. And so the parents were like, okay, we put your kid into remission instead of going to hospice. That's great. Next step is transplant. And they basically said, get away from me. She looks pretty good. Are these CAR T cells still in her body? Yes, they are, and they are there 10 years later. And they said, let's just forget that transplant. Let's see if these things will actually provide long-term disease control. And they have. So when we hit 5 years with her, Dad's asking me, well, is she cured? And I'm like, it's usually when I say patients are cured, but this is a new field of medicine. 10 years now, what else do we call it? We call it a cure. So this is actually our patient. I can use her name and her visage because if you Google CAR T, she's the first thing that comes up. The young lady next to Al Roker on the Today Show with her parents, Tom and Carrie Whitehead. Now 10 years later, 17 years old, getting in her senior year in high school, and never having any therapy beyond her CAR T. This then led to the registration trial that Nancy mentioned. And so this was these CAR T trials are all single-arm trials. They're registration trials, but they're not phase 3 randomized trials because there ain't nothing to randomize them to. Eventually, the FDA is going to say something about that, but right now they're not. And so we basically, 25 centers, 11 countries, cold chain across 2 oceans, the Atlantic and the Pacific Ocean. Didn't know we could do that. We could. Pharmaceutical manufacturing, first time a pharmaceutical company is making these cells instead of an academic GMP lab. Didn't know we could do that. We could. Did we get the same outcome for these patients? Yes, we did. 82% CR rate, 60% maintenance of remission at 2 to 3 years. And so the key question was safety. Some of these patients get super sick. 12% of my patients still go to the ICU. Which is down from 40% a few years ago. So we've gotten better at it, but it still can be rough for patients with a lot of disease. But we could do this safely. Not a single patient died of any treatment-related complication. And I had the experience of working with this great group of doctors, being on call 24/7 for 2.5 years straight to the entire planet, to which my wife said, okay, that's the only time you get to do that. So we've heard about issues of manufacturing, and in cell therapy, manufacturing is so complicated. It is a near miracle in my mind that we have been able to transfer the academic GMP manufacturing process developed by Bruce Levine and others at Penn out to a drug company that was the first time that somebody actually made cells at a pharma company. And this is actually the CVPF, which is what I would regard as a customer of theirs. I have nothing to do other than get cells from them. A best-in-class academic cell manufacturing, but it's highly, highly FTE dependent, it's non-mechanized, it's non-automated, and it takes somewhere around 11 or 12 days to make these cells, and then another week or so to qualify them, so over a 22-day total cell manufacturing process. And so these guys in there, I agree with the comment that was made that this is not a job opportunity for many people, but it's not a job opportunity for folks with a high school level of training. They need a lot more training than that. But this is what we want. We want to be training people to be sitting at that hood. And then we want to be training enough people so that the second we train one of those folks, they don't get pulled right out to one of your companies. And in one way, we're delighted with that, but in another way, what's the pipeline to provide folks who can do this and who can work in a bunny suit all day? It's really important. But another approach to this is to automate it, right? That's what drug companies are for, right? They should take this bespoke, one patient, one lot, 3-week, it's amazing that it works, process and turn it into something that's automated. There are these closed systems, the Multani Prodigy system, Lonza has a Cocoon, and these are systems that we're starting to use to see if we can make these cells in an automated fashion faster, maybe in just a few days. Maybe at point of care, certainly at a big academic center you can do point of care. Can you do it in a community hospital? You ask me, I say no, but I could be wrong. Not right now. And rapid manufacturing is helpful for two things. It gets you a product back to the patient faster, that's great. But nature made T cells to be inside your body, not outside your body. So 11 days is better than 6 weeks, which is what we used to do, but maybe 3 days is even better. So outside the body short, inside the body as quickly as possible, finish the manufacturing inside the patient, and of course people are now talking about giving vectors and making the cells from the beginning in the patient. We're not there yet. So this is really where this needs to go. I want to make a quick comment now to get away from T cells. So this is our experience with transplant at CHOP, Pediatric Hospital. This is not an adult thing, it's a pediatric thing. And so red is non-cancer and blue is blue is cancer and red is non-cancer. And what you see over time, the dates didn't show up, but we're basically going in decades, that more than half of the transplant patients that we treat don't have cancer. I'm an oncologist, what the heck? And a lot of those patients have hemoglobinopathies, diseases like sickle cell disease. And this is a great story because, and again, I don't have a lot of time here, but we have known the molecular basis for what's wrong in sickle cell patients since 1949, which is quite a while ago, like 10 years before I was born. Linus Pauling, brilliant guy, figured it out using tools available at the time, which is unbelievable. Now, of course, these sorts of questions are really easy to answer. That information did nothing for sickle cell patients for multiple decades. So, when I was a fellow in the 90s, early 90s, we still had oxygen, morphine, water for treating sickle cell patients with pain crisis. And so this now has led into the era of gene therapy using engineered stem cells. Cells, again, collected from the patient, created outside the body, brought back to the patient, single lot, bespoke manufacturing, because that's the only way to do this. Gene therapy for diseases like beta thalassemia, which is related to sickle cell disease, and sickle cell itself. And so this is the initial Bluebird paper about curing patients with beta thal with this autologous gene therapy process. And now, with all the interest in CRISPR, the Vertex company has developed a CRISPR gene editing approach to modifying stem cells to make them turn the patient into a patient that doesn't have beta thal, doesn't have sickle cell anymore. And this is also probably a year away from FDA approval, or at least for submission for a BLA. What happens in terms of approval, you can never predict. And this is the sickle cell study is a study that I'm honored to lead the committee for. However, of course, we now know Bluebird had an amazing back-to-back, and I have to give them full credit for this, back-to-back presentation in front of an FDA advisory committees, one minute to end, and I'm right there on my last slide, so thank goodness. And what they did was, the first day they presented on thalassemia, and the second they presented a completely different rare disease, doesn't matter what it's called, it's adrenoleukodystrophy. And in both cases, and for those of you who followed the Bluebird saga, I know nothing about business, I'm a pediatrician, but Bluebird was having a lot of trouble, but this is, despite this, both unanimous recommendations for approval, and one of these products, the thalassemia product, is now FDA approved as Integlo just about a month ago, and CHOP is set up to be their first qualified treatment center. So we're very excited to be delivering this therapy. The choice for these patients is this, maintaining their chronic disease, or getting a transplant from another person, which is something we do all the time. That's part of that non-malignant group that I showed you. But the reality is that getting cells back from yourself, even if they're genetically engineered, is infinitely safer than getting cells from another person. There are toxicities that we can totally skip. There's a 10% likelihood of dying during a transplant that is really the irreducible minimum these days for a transplant, and that's actually way down from what it was. And so autologous transplant is the way to go, in my opinion. However, I already mentioned this. What's the price of this stuff? $2.8 million. Now, I'm not in a place to say what's value from a pharmaceutical point of view or from any of these kind of financial engineering point of views, but we'll see how the market tolerates that. Our previous high was Integlo at 2.1. Here we are at 2.8, and we'll see how this goes. And it may be that the first folks at Vertex, which I know nothing about their commercial strategy or their pricing plans or any of that, they would never tell me any of that sort of stuff. But the bottom line is that really where they're going to go? Now, I will say that Bluebird has built a outcome-based agreement, and I'll end with that thought, that we can at least say, hey, if this works, great, and if it doesn't work, then we have a way to recoup some of that price that you paid for the product that's no longer working for an individual patient. And they can do that with some confidence because the vast majority of the patients have long-term success, which looks like a cure to us. So hopefully that's the way things go in the future, but anybody who's in this business knows that this is incredibly hard to do. Outcomes-based agreements are really difficult to negotiate and difficult to actually figure out how they should work. And really the only other one is for Kymriah and pediatric ALL. All the other CAR Ts don't have one that I'm aware of. So this cost challenge is the challenge for the field. This personnel challenge is the challenge for us right here. How are we going to train these people? How are we going to make enough of them? How are we going to be able to manufacture the cells? And a lot of these cells are actually limited by their manufacturing capability. And the final thought I'll leave you with, as companies get into the practice of making cells and running clinical trials and getting stuff FDA approved, is that better manufacturing and better execution beats a clinically better product every time. That the execution is the hard part. It's not even all this stuff. All this whole 10-year journey is not the hard part. The hard part is actually executing on manufacturing and getting it out to patients, which is still a solved problem, but not a completely solved problem, and something for us to be paying attention to for the next 5 years. And on that note, I'll thank you all very much.